Welcome. This is Dr. Ralph Esposito with Diagnostic Solutions Laboratory, and today we're going to be looking at the Sniff Navigator application within GenomicInsight powered by Opus23 Explorer. The Sniff Navigator is a particular application which you can find here on the left-hand panel, which you can decide to choose during your auto-curation process. The Sniff Navigator is basically known as a search engine for all of your DNA or your genomic data. So we can see here on the top right-hand side, you can click on this search bar and you can type in any information and the program will automatically search within all of the genes that have been reported by the program for your specific keyword. Now, you don't have to look just for keywords. You can also look for a particular gene. You can look for a particular SNP if you have it, or if there's any type of specific information that you have, you can insert that and it will curate and search the whole program. Let's take a look at each individual column. So we see here on the left-hand side, we have the curation. And this tells us, has this been curated or do we want to curate it for your particular report? Now remember, this is a auto curation, so you have full control over what you want to add or not. Secondly, we have the gene. And the gene just tells us where along the chromosome or the DNA, what is this gene that is associated with this particular SNP. So as we have discussed in prior videos, you can see that a SNP is a particular location or an allele within a gene, or basically it is the address of a SNP or an allele located on a particular gene. So you can see here the SNP for this gene, the VDR, which is the vitamin D receptor, is RS1544410. So it tells you that this particular SNP, this is the address on this street or on this gene. Next, we have specifics, and it tells us any specific information beyond the SNP number and beyond the gene that might be associated with this particular SNP or this particular gene. So we can see here in this additional vitamin D receptor SNP, it's associated with TAC1 or other specific information that might be associated with this SNP or this gene. Next, we look at the risk. The risk tells us what is the risk allele. This does not mean that it is a risk, or it does not mean an increased or decreased risk, but what it does show you is what is the interest allele that we are concerned about or that is of interest to us and that has been associated with the data and the literature. This may be different from the wild type or the normal variant. So the risk allele tells us what is the allele that is of interest. The type will tell you what is the risk or benefit of this particular allele. So this again is not saying that this individual has a risk. What it is saying is this allele, this interest allele, which is G, is a risk allele, which is something that might be alarming or something to be concerned about, compared to B, which would be a benefit. So if the individual had the T allele, it would be a benefit for them. Now, we look at what is the actual information for this individual. So we see that this client has two A alleles for this particular SNP, does not have the homozygous G alleles, which does not put them at risk, which means that they are unlikely to be at risk for issues that are associated with HLA, also associated with post-Lyme disease syndrome, AS, which is a type of autoimmune condition, or other autoimmune conditions that might be associated with HLA-B27. Next, we look at the magnitude. Now, the magnitude tells us how much data is supported by this particular SNP located on this gene. So this is the overall gene function to have an effect on disease risk. And this is something that is an important thing to pay attention to because this is an algorithm that is decided based on the data that we have, how important this is for an individual's health. Now, things that can decide the magnitude would be how many studies. Are they animal studies? Are they in vitro, in vivo, or are they human studies? And by putting all of these together, the program will decide the magnitude of this is in 10, or the magnitude might be even lower, such as a 1. This is something that we take into consideration when we want to curate an actual SNP because it tells us how much of this is clinically relevant. The more data and the more literature that there is to support a particular SNP, especially if it is a risk type allele, then the magnitude would be something to pay more attention to. Next, we have the effects. And this is something new to Opus23 Explorer. And this tells us what impact it has on the gene function. So we can see here a green arrow increases gene function, whereas we can search for a downward arrow, which is black, and a black down arrow will completely stop gene function. And then next, we can look for a red downward arrow, which decreases gene function. So green would increase function, red would decrease function, and black would completely stop gene function. And then in the last column here, we have keywords. Keywords is basically how we parse the information and connect it to particular conditions, or disease states, or even certain nutrients. So for example, if you typed in vitamin D, these are all the conditions that would be associated with vitamin D, or if there's some type of correlation. Now you can see here, there's several genes in here that have been associated with vitamin D. But if we wanted to look something even more simple, we could even search MTHFR. And if we search for MTHFR, we can see all of the genes and SNPs that have been associated with MTHFR. Not just the MTHFR gene, but the MTRR and CBS and SLC19A1, which can be utilized and informs the practitioner of other things to pay attention to. Now, beyond just searching the particular gene, such as COMT, the search results then allow you to click on that gene, which then gives you more information about the gene or the particular SNP. Take, for example, COMT, which is catechol O methyltransferase. Clicking on the gene will then give you more information about the particular gene. So what we see here is that this is a very detailed description of COMT, which is designed for the practitioner. However, if you click on the Show Client description, it breaks it down into a very simple form, which will be described and shown to the client in their curation report. They will not see the detailed version below, as that may be confusing to them. On the top left-hand side, you can decide if you'd like to curate this to your personal curation, and then it gives you a percentage of how many users are now curating COMT into their curation reports. And it shows here about 50% of individuals are curating COMT into their reports for clients. So that tells you that this might be an important SNP and very common SNP to be incorporating in your reports. As you scroll down, we then get more information about this particular gene, and it gives you different variants, and it tells you exactly what they may be doing. Furthermore, it does provide the references for everything described in the above description. Then we see where COMT may exist in other macros or in other algorithms, and we see that it does exist in the B12 supplementation, impacts on placebo, other impacts on placebo, and then risk of chronic fatigue syndrome. And it will tell you adjacently whether that is true or false. These also can be clicked on, and it will link you directly to that macro, whether you like to curate it or not. Then we see the protein-protein interactions, which has a lot to do with how these genes and the byproducts of these genes are interacting with other genes and their byproducts. Then we see the results for this particular client and what SNPs that they have. And as you can see, there's many different SNPs for COMT beyond the simple 2 or 3 that are reported by some programs. Moving down, we then can explain other PubMed references that are referenced, particularly for COMT. And you can see here we have about 31 different references for COMT, which is all incorporated into the GenomicInsight program. As you scroll down, we can then see different agents that are impacting COMT activity. Green symbolizes an agonist, so something that may improve or increase function, and the orange may indicate something that decreases function. And the farther out that the agent comes in the pie graph here, the more evidence or more research there is indicating that that might be an agonist or an antagonist. So therefore, we see that magnesium has a significant amount of data showing that it might be an agonist or improved function of COMT, whereas EGCG might do the opposite. As we move down, we can then view each one of these agents, and it tells you whether the action is antagonist or if it's an agonist, and then some may be undefined. And in association with that, you'll see the Pubmed reference, which can be clicked on, and that'll tell you and show you the actual study. If you continue to search all of these different agents, you can then click on the Pubmed article, and the Pubmed article shows up directly there and you get an abstract. Therefore, not only is this evidence that this data is peer-reviewed and is evidence-based, but also it gives the practitioner a little bit more confidence knowing that what they're doing is based on actual literature and not something that has been reported by a potential theory or by another practitioner, which could be true or untrue. Again, this will then show you other genes that have been associated in this actual article. The practitioner can then take this PMID and utilize that to read the full article on PubMed. Exiting out of this will then bring you back to the Sniff Navigator, and you can start over again looking at other genes that have other information, getting the short descriptions, and furthermore showing the same information that we've seen for COMT, showing all the different studies and the different agents that may have an impact. Additionally, the practitioner can also click on the SNP. By clicking on the SNP, you get actual data for that particular SNP, not the whole gene, which again is more of the particular single nucleotide polymorphism or the actual address of that mutation on the gene. So you can see this one here, this particular SNP is associated with VDR. We see that it has a power factor of 7, the evidence basis of 5, and the curation level of 5. The power factor is decided by an algorithm within the program which takes into consideration all of the various different studies, including in vitro, in vivo, and human and animal trials, to then curate an actual algorithm showing how important or how influential this SNP may be. So as we move down, we can see there's different agents that activate this particular SNP. So you can get SNP specific, which also does give you more agents here, and it'll tell you adjacent to that whether it is an animal trial, a human trial, or something that has been unidentified. This will allow the practitioner to understand how robust the data might be for that particular agent. And as you can see, many of the vitamin D trials have human studies, which may give the practitioner a little bit more confidence in knowing what this may or may not do for that particular SNP or that particular agent. And lastly, we see if there are any algorithms or macros that may be associated with this particular SNP, and currently there are none. Again, we can exit out of this and return back to Sniff Navigator and begin the whole process again. Thank you very much. This was Dr. Ralph Esposito with Diagnostic Solutions Laboratory, and this was the tutorial on the Sniff Navigator in GenomicInsight powered by Opus23 Explorer.